This tutorial describes how the three non-methylcellulose instrument-based assay kits, HALO, HemoFLUOR and HemoLIGHT, are set up for blood-forming stem and progenitor cell research. The primary tool used for research of the blood-forming system since 1966 has been the methylcellulose colony-forming cell, CFC, or colony-forming unit, CFU assay. To improve reliability, reproducibility and consistency, as well as ease of use and more rapid results, the methylcellulose was replaced with suspension expansion culture, or SEC technology. The HALO platform was the first to incorporate SEC technology, and in doing so, elevated hematopoietic research to a level of reliability, reproducibility, and rigor that has not been seen in over 50 years. HALO was more recently followed by HemoFLUOR and HemoLIGHT. All three platforms use 96 well plates and are, unlike the CFC assay, all instrument-based assays, which means there is no subjectivity at all. HALO incorporates an ATP bioluminescence readout, while HemoFLUOR incorporates a fluorescence readout, and HemoLIGHT uses an absorbance readout. Although the CFC assay has been a mainstay in hematopoietic research for over 50 years, it is well known by everyone who has used the assay that it is certainly not the most reliable. It is usually only used to count and identify colonies as being derived from a particular cell population. The results and interpretation are therefore quite limited. Despite this, the assay's use has sometimes been raised to a level that cannot be scientifically grounded. In addition, claims have been made that the CFC assay can actually do more than is physically and scientifically feasible. For example, it has been claimed that the CFC assay is a proliferation assay. Cell proliferation is required to form a colony, but unless the size of the colony can be measured using an accurate and calibrated method that provides a quantitative cell number for the colony, the CFC assay is not a proliferation assay. This is made particularly difficult, if not impossible, since most colonies do not conform to a regular shape. In fact, they are quite irregular in shape. Add this to the fact that to identify a colony, the cells have to differentiate and mature. This takes time. That is why colonies are counted when cell proliferation is actually decreasing or has ceased altogether. It stresses an important and often forgotten basic scientific fact. Proliferation always occurs prior to differentiation, and the differentiation process occurs by default under normal conditions. Only under special circumstances, such as those used in the Cameo 96 assay from preferred cell systems, can cell proliferation in colonies be determined. But even then, two different endpoints must be used. Just as with the CFC assay, it is the growth factor cocktails that define the population being measured. However, since no colonies are formed from colony-forming cells, we use a slightly different nomenclature to define the different cell populations detected with assays that incorporate SEC technology. For stem cell populations, we prefix the population with the letters SC. We use the letter P to show a progenitor cell population. For HALO, HemoFLUOR and HemoLIGHT, there are individual assay kits for up to 6 different stem cell and 7 different progenitor cell populations, available for either human, non-human primate, horse, sheep, pig, dog, rat or mouse. In addition, Preferred Cell Systems also offers global assay kits in which 4, 5 or 7 stem and progenitor cell populations can be individually assessed simultaneously. Regardless of the kit you have purchased, make sure that the contents of the assay kit correspond to the packing list included. If there are any discrepancies, please let us know immediately and we will try and rectify the situation. Store the assay kit in a freezer at -20 degrees Celsius. To set up an assay, you will need to remove, first, the master mix, which contains everything needed to grow the specific cell population you requested. No other additions to this master mix are necessary. Second, you'll also need to remove the sterile, individually wrapped 96-well plate. If you purchase the Halo kit, the plate will be white. If it is a fluorescence kit, the plate will be black. An absorbance kit has clear plates. Everything else can remain in the kit box and in the freezer. The technical manuals can be found on the Halo, HemoFLUOR or HemoLIGHT web pages of the Preferred Cell Systems website. To set up either Halo, HemoFLUOR or HemoLIGHT, you'll need the following supplies. First and foremost, you'll need the target cells. For research assays, these can be any hematopoietic tissue or organ. With the exception of embryonic cells and fetal liver cells from rats and mice, all tissues should be fractionated to a mononuclear cell fraction prior to use. You'll need sterile 5 ml plastic tubes with lids, single channel pipettes with sterile tips. You won't be needing any syringes and needles for this process. You'll need a vortex mixer and a humidification chamber, the details of which you can find in the technical manual. And finally, a humidified incubator set at 37 degrees Celsius and gas with 5% CO2 and, if possible, 5% O2. The low oxygen tension reduces oxygen toxicity and improves plating efficiency. Once you have everything on hand, you're now ready to set up the assay. The procedure for setting up these assays is the same regardless of whether you're using Halo, HemoFLUOR or HemoLIGHT. Let's now start the setup procedure. We should be setting up cultures for 2 cell samples, so 2 5 ml sterile tubes will be labelled 1 and 2. Don't forget that you don't have to use the whole plate. Included with the kit that are sterile adhesive foils that keep unused wells sterile. Next, we have to prepare our 2 sample cell suspensions. These will be samples 1 and sample 2. The working cell concentration based on the mononuclear cell count is set at 500,000 cells per milliliter for each sample. This means that the final cell concentration will eventually be 5,000 cells per well. We are going to set up 6 replicate wells per sample. We will now dispense 0.585 milliliters of the master mix into each of the empty tubes. If we only wanted 4 replicates, we would dispense 0.405 milliliters of the master mix. And if we wanted only 2 replicates, we would dispense 0.315 milliliters of the master mix. Once the master mix has been dispensed into each tube, we won't be needing it anymore. Gently mix cell sample 1 to resuspend the cells and transfer 0.065 milliliters into tube 1. This volume represents 10% of the final volume of 0.65 milliliters, which will be sufficient to prepare 6 replicates of 0.1 milliliters each. With this step, we are diluting the cell suspension tenfold, so its concentration in the tube is now 50,000 cells. If you were preparing only 4 replicates, the cell volume would be 0.045 milliliters, and for 2 replicates, 0.035 milliliters. Now gently mix the second cell sample and dispense 0.065 milliliters into tube 2. We will not be needing the 2 tubes containing the cell suspensions. These can now be removed. The 96-well plate can now be removed from the envelope. It is worth noting that for bioluminescence, white plates are used. For fluorescence, black plates, and for absorbance, clear plates are used. The next part of the procedure involves dispensing 0.1 milliliters into each of the 6 wells of the first and second row of the 96-well plate. This will correspond to sample 1 and sample 2 respectively. This will reduce the cell concentration another 10-fold, so that the final cell concentration per well will be 5,000 cells. It is important to dispense the culture master mix into the center of the well, not on the walls of the well. Once the 6 replicate wells of sample 1 have been prepared, repeat the process for sample 2. Replace the lid onto the 96-well plate. Transfer the 96-well plate to a humidified container. This is described in detail in the technical manual. Then transfer this to the humidified incubator set at 37 degrees Celsius with an atmosphere containing 5% CO2 and, if available, 5% O2. The low oxygen tension, which is approximately 45 millimeters of mercury and also approximates the venous oxygen tension, will help reduce oxygen toxicity and increase plating efficiency. For animal cells, incubate the plate for 4 days. For human cells, use 5 days of incubation. So let's briefly discuss the results you might get. Some of the questions people ask are, how can you count colonies? Or how can we identify the cells? Or how do we know we're getting the same cells produced as in a colony? The first thing to remember is that for neither HALO, HemoFLUOR, nor HemoLIGHT are you going to count colonies, because no colonies are produced. The growth factor cocktails would be similar to those you might use in a CFC assay. So you're going to get the same cells produced whether they are grown as colonies or in suspension. And the evidence for this can be established using flow cytometry. This can all be a little confusing, especially if you have never used an instrument-based proliferation assay. But don't despair. You'll find that HALO, HemoFLUOR, and HemoLIGHT are not only very easy to use, but provide you with results that are easier to understand and have much more meaning than any colony counts. Instead of detecting whether cells can differentiate, you are asking the question, will the cells be able to proliferate? And if so, how much can they proliferate? It follows that if the cells can proliferate, the cells must be viable. Therefore, HALO, HemoFLUOR, and HemoLIGHT assays are also metabolic viability assays, and as such, viability is not measured as a percentage, which is the case for dye-exclusion viability methods. Instead, viability is measured quantitatively as relative luminescent units, or RLUs, for HALO, relative fluorescent units, or RFUs, for HemoFLUOR, or absorbance units for HemoLIGHT. When cells proliferate above a specific amount, you will know that the cells are viable and can sustain proliferation. For HALO, the acceptance-rejection level for hematopoietic cells to be viable and sustain cell proliferation is 0.04 micromolar of ATP. This is described in more detail in the HALO technical manual. For HemoFLUOR and HemoLIGHT, this level must be established empirically by the investigator and can be easily ascertained by comparing dead cells with live stimulated hematopoietic cells. For HemoFLUOR and HemoLIGHT, it is good practice to include a cell dose response to normalize the results. This allows you to compare results between experiments at any time. Normalization is not necessary for HALO because this assay includes controls and ATP standards that calibrate and standardize the assay, allowing you to compare results at any time. With any of these assays, you can measure 2 types of hematopoietic cell proliferation. The first is proliferation ability. This is the amount of proliferation that occurs at a specific time point and at a particular cell concentration. This is a single point determination and is used in most experiments. The second type of proliferation is proliferation potential or capacity. This is determined using a minimum 3-point cell dose response and, as you see, includes proliferation ability. However, the proliferation potential is measured by the slope of the cell dose response. The steeper the slope, the more primitive the cells being measured. This is how you can distinguish a primitive stem cell population from a more mature stem cell population. You can also determine how pure a cell population is by measuring its proliferation potential. There are numerous applications where measuring proliferation potential is of the utmost importance and therefore cannot be performed using a CFC assay. This concludes the tutorial. If you have any questions or comments, please contact us and we will get back to you as soon as possible. Thank you.